the Clinical Development Program for PMM2-CDG.
Great, thanks, Andy. And thank you to Karen and the organization of the CDG CARE to allow us to present our data today on our studies and where we've come from and where we're going. So as Andy said, my name is Marino, for those who haven't met me. I'm the Chief Medical Officer here at Glycomine, and I'm a pediatric endocrinologist by training. So this morning, and I'll try not to go over it all because I know I'm probably the only thing standing between us and lunch, but we'll just do a very quick intro. I know we've talked a lot about PMM2-CDG this morning and CDGs in general, but I'm just going to set the stage as to how GLM-101, the therapy we're looking into as a potential treatment for PMM2, fits into that disease milieu of treatment. Then we'll go into a little bit of the development of where we've come from in terms of getting to where we are today, which is in our 2B pivotal study, and how the natural history study has also helped us understand endpoints and being able to compare and move things forward within the program. And finally, we'll end with where we are in the clinic at this time and share where we expect to be by the end of the year, hopefully be able to share even more results into 2027. So this is a very simplified version. I know we've seen lots of more complicated diagrams of glycosylation, but as we've been talking about over the past several days, glycosylation is a very important part of biology. If our proteins are not glycosylated, they do not function as they should. And in terms of PMM2-CDG and other CDGs, if the proteins are missing their glycans or they have abnormal glycans put on their proteins, they won't do the jobs they need to do, and it translates into the disease state and the phenotypes and the symptoms that the patients are experiencing. So very simplified pathway. So what does PMM2 do? It takes the upstream substrates and allows that pathway to move forward. So in particular, we highlight the mannose-6-phosphate needs to be converted to mannose-1-phosphate. Very simple. The phosphate needs to move from the 6 position to the 1 position. But if it can't do that, it becomes a block, and the rest of glycosylation cannot happen. So what are we trying to do to fix that? Well, with GLM-101, we're trying to fix it by giving back the cell the substrate it needs. So the GLM-101 here is a liposomal product of mannose-1-phosphate. So what we're trying to do is be able to get mannose-1-phosphate into the cell so we can bypass the block of PMM2. And the trick of GLM-101 is that it is a liposome, because if you would just give a patient mannose-1-phosphate, within 4 minutes, that mannose-1-phosphate would just be mannose. So you have to protect the mannose-1-phosphate, and you have to allow it to get into the cell. And by encapsulating it into a liposome, so a fat-like particle, and these fats are really what our body already knows and makes, it's not a foreign substance, the drug is able to accomplish that goal. So that's just the basic biology. GLM-101 is a substrate replacement. I know we've heard a lot about gene therapies and RNAs, but this is really a very simple concept of replacing back what the cell needs. So I'm going to switch gears a little bit, and we'll talk a little bit about clinical trials in general, as well as how Glycomine has moved through the process of the clinical trial development program to where we are today. So just to remind everyone, and this isn't to GLM-1, this is the typical pathway of what clinical trials, typically the road that is taken in order to get to an approval. But what you're not seeing on this stage is actually years of preparation. So we've talked about drug candidates and developing new therapies. Well, before you even get into being able to put something into a human being, you have to test it. You have to test it in zebrafish. You have to test it in worms. You have to test it in animal models, in fibroblasts. And then after you think you have a drug that could be useful, then you have to make the drug. And you have to make it in a way that's safe for human beings to be able to take it, which the bar is higher than giving it to a mouse or a worm. So all that development takes years, even prior to getting to that first phase 1 study. into a healthy volunteer. But what's important for rare disease in general is to have natural history studies, and Dr. Lamb's going to talk about that tomorrow, so I'm not going to go into that too much. But natural history studies are needed in rare disease when nothing has been developed yet in terms of endpoints and outcomes. So there's been a few natural disease studies. Glycomine, we also had a natural history study that looked at PMM2 over 5 years. And some of that data we'll share today as we look at it in the context of our treatment study. But after we started our natural history study and we did all of that development prior to, as I just discussed, we went into what's called a phase 1 study. So a phase 1 study is you give it to healthy volunteers in order to figure out, well, what dose should we be bringing into patients with the disease, and generalizing how well it's tolerated and how it's metabolized in the body. And once you have that data, you can then say, okay, we think this is the dose that would be safe that we would start with in patients. And that's where you come into what we call a phase 2 study. So we've completed our phase 2A study, so you may hear some jargon on, well, a phase 2A versus a phase 2B versus a phase 3. So a phase 2, usually you start with a small number of patients in the disease. You start with some exploratory endpoints to see how the drug is affecting any sort of efficacy markers, how the patients are feeling. You look at safety, you look at how the drug is being taken up in the body, and you measure it in order to see how high it goes and when it actually becomes eliminated. So you're getting just more information. And you're also, again, looking at those endpoints in order to bring that into your, what we call, pivotal study. So that could be a phase 2B, which is a bit of a larger study, or a phase 3. And if you've gone through all of that and you show that you have not just an efficacious drug, but a safe drug, then you can approach regulators. I have FDA on here, but it's FDA, it's EMA, it's Health Canada, it's Australia, it's Japan. You have to go to each individual country, MHRA, because I know we have UK folks in the audience. You have to go to each individual country and ask them for approval. So you do this in a way that you hope that the information that you develop will be acceptable to all jurisdictions, because some jurisdictions want different things. So keeping that in mind, you try to develop your studies so it really can be global at the end of the day. So what have we done? So like I said, we've already, we're at the end of our natural history study. I know there are some people in the audience who may have been part of that. I know there were some clinicians. It was 5 years, and I think we're finishing up this month, actually, after 5 years of this study. Our phase 1 was done several years ago, which led us into our phase 2A, which we completed in November of 2025. So that's the data I'll show you today. It's no longer preliminary, it's final data in the 2A, which has given us the information and the knowledge to bring into a phase 2B, or what we hope will be a pivotal trial that will lead to that final ability for clinicians to be able to take out their prescription pad at the end of the day and prescribe a medication that would help patients with PMM2. So the phase 2A that I'll talk about that we had just finished, I'll give you an overview just very quickly of the design. So open label, that means everybody knew they were getting GLM 101. We knew it at Glycomine, the clinicians knew it, the patients knew it. It was 6 weeks in length, and it was weekly, because our dose is a weekly dose of GLM 101, and it has to be given intravenously. And we looked at doses between 10 and 30 milligrams per kilogram, so it's a weight-based dose. So depending on how the weight of the patient depended on how much GLM 101 they would receive. We enrolled 27 unique PMM2 patients, and I put up there the N of 15 of ICARS, which is the primary endpoint and the data that I'll show you. And we actually dosed all the way down to age 2. But we couldn't do that from the beginning. We actually had to start with adults, and then we dosed adolescents, and then we dosed older children, and then younger children. Because as we developed our knowledge and our understanding of the safety, we then brought it into the younger population. As I said, the primary endpoint was ICARS, and that's the data I'll share. And of course, you always look at safety. Again, the PK is how the drug looks in the body after you dose it. And we looked at other endpoints. So I know we keep talking about ICARS, but there's other ways and there are other things we can measure in order to see how patients respond to therapies. So I'll give you a little bit of information in our data on something called the GROW. So this is an endpoint that's never been used in PMM2. It's been used in spinal muscular atrophy. But we have to look at other diseases and what other what other trials have done to gain knowledge to see, well, if they're using this, would this be useful for PMM2 versus trying to develop things that are brand new? So not having to reinvent the wheel with scales that are out there, but seeing if we can leverage what others have already done also helps move development a little bit quicker. So in terms of safety, it was very well tolerated. We had no serious adverse events. All of our adverse events, which are any medical issue that happens to a patient, it could be a cold, they could fall down and fracture a foot, everything has to be reported. And we gather all of that data and we look at it in totality. And we ask the clinicians if they have something happen with their patient, was it related? Do they think it's related to the treatment? Or is it just, that's just part of life, everybody gets a cold? And then we look at the data in that respect too. And we say about half of our patients experienced an adverse event that was thought to be related. I could say for the majority of patients, they did very well. The most common one we saw is something called an infusion-associated reaction, which we actually knew was a very high potential in the study because we saw it in our animal models, we saw it in our phase 1, and we were able to also then kind of warn the clinicians. We actually warned the patients in informed consents. So we knew to be able to be on the lookout for this. And as we would have predicted, it happened in about a third of patients. It only occurred in the first few infusions, and after that, patients tolerized to the GLM-101, and we didn't see it after that. So moving on to efficacy. So our primary endpoint in the study was ICARS. So that's the International Cooperative Ataxia Rating Scale. So we might have heard a little bit in talks yesterday about the ICARS. It's something that's been used, again, in other registrational studies. It's in the PMM2-CDG guidelines now, so it's pretty well accepted in the community. And it's assessment by your clinicians or your physical therapists to assess the ataxia in patients. And what's special about ICARS versus some of other ataxia scales, and there are several out there, is that it hits on 4 different domains that are relevant to patients with PMM2. So it has an eye domain, it has a speech domain, it has a gait posture domain, and the kinetic function domain is basically a domain of how shaky patients are and how steady they are. So when we look at the data from our study, what we're seeing here on the y-axis is change from baseline in ICARS score. So with ICARS, the higher your score, the more severe you are. So you want to actually see things decrease, so going negative. And here we see we looked at patients at baseline, at week 12, and at the end of the study at week 24. And the light green lines, those are individual patients. So that's what we would call a spaghetti plot. So you can kind of see, people can present averages, but this is real data for each individual patient. And on average, by the end of the study, we saw about a 10-point improvement in ICARS. And this was pretty astounding to our clinicians. They, when we started looking at this data, they were like, we've never seen improvement in ICARS before. Because usually, as we learned from our natural history study, patients with PMM2-CDG, they either have a little bit of worsening, but for the most part, it's fairly stable. And this understanding is actually critical to put into context what we're seeing in the study. Because if our natural history study said, well, these patients are already getting better, then we would say, well, this was going to happen whether or not GLM-101 was given to these patients or not. But that's really not what we were seeing through the testing that we've done. So just quickly, the GROW is a brand new assessment for PMM2. And the reason why we started looking at this is because, as we heard yesterday, ICARS can't be performed by all patients. You have to be cooperative. You have to understand what the clinician is asking you to do. And so how do you measure that, in patients that are too young or autistic and cannot follow those commands? What else can we be looking at? So this is a scale that was developed by Nationwide Children's Hospital for spinal muscular atrophy, and it's by observation. And it really assesses the whole body strength, motor development, and it's across all ages. They looked at it down to age 6 months, and really in those diagnosed with neuromuscular disease. So we put this in the protocol just to see, and understand from the clinicians, is the scale working? Is it good for PMM2 patients? And are we seeing an effect? So here you have to flip your brain around because it's still a 100-point score like ICARS, but a lower score means greater impairment, versus with ICARS, a higher score means greater impairment. So what we saw are trends. So we've never done this before, we've never looked at it before. So we saw in the patients after we added this into our protocol, had about a 5-point improvement over the 24 weeks. So no control data, so I can't put this against natural history study. We didn't do this in the natural history study. But this shows that patients look like they're improving in terms of their gross motor function. And then clinically, we ask patients and we ask clinicians, how are you doing? How are you doing overall? Are you feeling better? Are you improved? Are you worse? Are you the same? And in this study, we were able to show that all the clinicians and every patient except for one who said they didn't have a change saw some improvement from before they started the study. But then we wanted to know, okay, well, that's generally how you feel. How do you feel in terms of your severity of your ataxia? How do you feel in terms of your severity of your motor function? And the clinicians all reported a reduction in the severe or very severe ataxia, as well as in the gross motor function for patients. And the caregivers also reported the same sort of improvements in ataxia and gross motor function severity. So I think I have 2 minutes left. So I'm just going to jump to our new study. And I guess it's not so new anymore because we're probably almost halfway done with the first part of the study called the POLAR trial. And what we were able to do is take our learnings from the 2A, saw this wonderful signal in ataxia and gross motor improvement, and really put it against the gold standard control, so that placebo randomized controlled trial paradigm. And that's where we are today. So we've enrolled the whole trial. We enrolled 43 patients with PMM2-CDG over the age of 4. They have to be able to perform the ICARS because that's our endpoint. And we knew in our phase 2A, the smaller kids couldn't do it. Some of the kids neurocognitively couldn't do it. So we had to make sure that we actually got the data we were trying to get at. Otherwise, you would need many more patients in the study, and you would need different endpoints. So we're looking at ICARS, and then again, we're looking at some of these other endpoints. The GROW is back in here, so now we'll have an understanding of what does GROW look like in untreated patients over 6 months. So we'll have that benchmark to compare, as well as some other exploratory endpoints from grip strength to neurocognitive function, again, always safety and pharmacokinetics. We are global. We are in 15 sites across the US, Europe, and the UK. And like I said, we're about halfway done with that first part of the placebo control before patients go into the open label. And we expect data readout in early Q4. So it's pretty exciting. And, it's basically the end of my talk, but we hope this is really the beginning of further studies and further understanding. And thank you to all the patients and the clinicians and my team, because without everybody pulling in, with a 15-site trial, you can imagine it takes a lot of coordination and a lot of effort. So, thank you.
Great. Thank you so much.